Hey guys, so we're going to start chapter 13 right now, and in this video we're going to go over section 13.1, which is all about translation. So I'm going to share my screen with you guys as per usual, and we'll get started. Okay. So, translation. So this is a translation of mRNA, and this depends on a few different things. There's a whole, I've been calling it the translation machinery, right? And this is just because there's a lot more stuff that we need for translation as opposed to transcription. So at this point, we're going to go from mRNA to protein. And at this stage, we're going to require the presence of structures known as ribosomes, which are basically the site of protein synthesis, right? Which is basically what translation is, it's protein synthesis. And we're also going to need the tRNA, transfer RNAs, which are essential in the sense that they're actually the ones that bring the building blocks of the proteins and for the protein synthesis to the site of protein synthesis, the ribosomes. So as you guys know, the genetic code stores information as triplet nucleotides in DNA. These triplet nucleotides are known as codons. And so these codons are going to be, are actually going to be made into mRNA. So they're going to be transcribed. This transcribed mRNA is what we looked at in chapter 12, where we saw that we got a complementary strand of mRNA compared to the template strand of the DNA of whichever gene we're actually transcribing. The final gene product of gene expression linear series of is a linear series of amino acids as prescribed by the genetic code. Okay, so we already looked at the genetic code. We saw that specific code. We have 64 triplets or 64 codons, and each one codes for a specific amino acid except for 3, right? The stop codons, the UGA, UAA, and UAG. Those 3 do not encode for an actual amino acid. So now we're going to be looking at how this mRNA and its codons are read, each codon one at a time. Each codon is coding a specific amino acid. So it's basically we add one amino acid one at a time as we synthesize this protein. This whole process is known as translation. So it's so the actual definition that your textbook gives you, it's a biological polymerization of amino acids into a polypeptide chain. Remember, a polypeptide chain is just a sequence. It's that linear sequence of an amino acid. We'll see later on that this linear sequence has to then kind of bend and shape and fold in on itself to give us a specific 3-dimensional protein, okay? So translation requires a lot of different components. First off, of course, we need our messenger mRNA. This is what is storing that information that we gathered from DNA, right? So this is where we have our codons that code for amino acids. We're also going to need to bring those amino acids into the equation. We're going to need a place for this to occur. That's going to be on the ribosomes. And then we're going to have something that brings the amino acids to this, and those are your transfer RNAs. Okay, so your tRNAs, we'll actually look into the structure of the ribosomes and the structure of your tRNAs a little bit. But just to give you an idea, just really quickly, your tRNAs are just structures that are able to bind to the mRNA codon. They're able to bind to the mRNA codon because they themselves have a ribonucleotide sequence on them known as the anticodon. So this anticodon is complementary to the codon, and this is how they're going to bind to the correct codon. And as they bind to the correct codon, depending on whatever genetic code, depending on the genetic code, they're going to bring the appropriate amino acid to the equation. So if they have an AUG, hold on, I'm actually going to draw this out. Bear with me. If the codon on our mRNA is an AUG, stop share. If this is my mRNA, right? So this is my mRNA. I have AUG, CCC. Can't remember any of the other codons, guys. Blah, 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 blah. Okay. My you guys know that AUG, that's our start codon, and that's going to encode the beginning amino acid, which is always methionine, right? It's also methionine in bacteria, except it's a methionine that's a little bit more kind of tweaked. It has a chemical moiety formo on it, so it's known as formomethionine. So our tRNA will bind on one side of the tRNA. It actually has the correct amino acid. So the tRNA actually looks like the 3-dimensional configuration of one. It kind of looks like an upside down L. And on one side of the L, it actually binds to the appropriate amino acid. In this case, it's methionine. So I'm going to write here MET, right? And then here, where it's going to bind to the mRNA, it literally physically binds this. So we have hydrogen bonding between the start codon, or one of the codons, and the anticodon on the tRNA. So here it's going to be If this is from 3' to 5', right, there's going to be an anti-parallel binding. So we have something like G will bind to C, U will bind to A will bind to U. Okay, so this is the anti-codon right here. As you can see, it's on the other side of, is on one side of the tRNA. The other side of the tRNA is actually bringing the methionine, the amino acid, to it. So when this binds to the codon, now our methionine, or the whatever amino acid we need, is going to be in close proximity to that site of protein synthesis. So the amino acid is actually brought to be added to the polypeptide chain. Okay, so that's basically what the tRNA is in a nutshell. We're obviously going to be going over the structure in a little bit more detail and talk about the ribosomal structure as well. So first things first is the ribosomal structure. Now the ribosome has an extremely essential role in this translation, right? This is where translation actually happens. It's the site of protein synthesis. And the ribosome is composed of a few things. It's composed of a few proteins, right? But the main thing, the actual catalytic part of a ribosome, and so therefore the part that actually allows for the ribosome to carry out its role, is going to be made out of rRNA. And there's a lot of or also known as ribosomal RNA. There's a lot of ribosomal RNA that constitutes and makes up the structure of the ribosome. This rRNA, because it is catalyzing, that means it's enzymatic, and so we call it ribozymal, right? It's like a ribosome. Ribosome, I said the same thing. Ribozyme, that's what I meant to say. It's like a ribozyme. It's a type, it's RNA, but it can work enzymatically, and this is what ribosomal RNA is. Ribosomal. I'm getting confused between the two words, sorry. So the rRNA molecules perform the catalytic function associated with translation. The proteins are basically there just to hold the things together, kind of like to, you know, keep the structure together and to kind of tune things up, to just perfect them, fine tune them a little bit. Okay, so they're not really directly involved with the process of translation. They're not really directly involved with the process of actually synthesizing the protein. That actual synthesis part, the creation of, if you guys remember how the proteins are made, there's amino acids are bound together by a peptide bond. So that creation of that peptide bond is spurred on by and carried out thanks to our RNA. That is the catalytic part of the ribosome. Ribosomes look like little snowmen, or like, in my opinion, they look like cheese, because we have a specific cheese in Italy that looks like them. And now I'm thinking of that again. Oh gosh. Okay, so ribosomes look like snowmen. We have a small part, a small head on top, and then a bigger body at the bottom. And so they have a large and small subunit. These large and small subunits are made independently in the nucleolus. We went over this in Unit 1. So they're made in the nucleolus if it's in a eukaryote, and then in the nucleolus it exits out into the nucleus, through the nuclear pores and into the cytoplasm, where the ribosome will eventually arrange and come together, fully assemble itself once protein synthesis happens. So the arrangement of the snowman is not how ribosomes look when they're not working. They only come and look like the large and small subunit assemble only once the ribosome is actually carrying out protein synthesis. So together, the large and small subunit make a single ribosome. We can also call this a monosome, but I have to let you guys know this really isn't a word that is often used. You can just say a ribosome. They're characterized on the basis of sedimentation, which is known as the Svedberg coefficient. So every time you see an S, right, after all these numbers, that is the Svedberg coefficient. And that's just basically the amount of, like, the sedimentation rate. If you imagine it going through a centrifuge, it actually goes through a sucrose gradient. If you basically centrifuge these ribosomal subunits, each of them, depending on how heavy they are, so depending on their mass, depending on their volume, and depending on their shape, right? Each of them has a specific Svedberg coefficient. So this Svedberg coefficient, basically, this whatever molecule you are looking at, it doesn't necessarily have to apply to just ribosomes. You can do it with anything, essentially, and everything is depending on the molecule's mass, the shape, and its density and volume. It's going to sediment in a specific place, right? So really, really heavy things are going to sediment down all the way down compared to really, really light things. Now, the thing to consider, though, with this Vedgar coefficient is that you're not just looking at the heaviness or the mass of the molecule, you're actually also considering the shape, right? So if we see this, the prokaryotic ribosomes, so ribosomes that we find in bacteria and archaea, have this Vedgar coefficient, so the total monosome of the entire ribosome, large and small subunit, is 70S. But when we only do the large subunit, it's 50S, and then the small subunit is 30S. And you may be thinking, Alessia, 30 plus 50 is equal to 80, is not equal to 70. You are correct. But remember, this Vedgar coefficient is also considering the shape and then the density of the actual molecule. So when the molecule is together, even though originally the large is 50 and the 30 is, and the small is at 30S, when you put them together they will sediment at 70S, just because you also have to consider that shape. The same can be said about eukaryotic ribosome. When you look at the large subunit, it has a 60S, 60 Vedgar coefficient, and the small has a 40. Together, they don't actually make 100, right? When you have the monosome or a full functional ribosome, it will sediment to about 80S. And you may not think this is a big deal, okay? Who cares that the bacterial ribosomes are 70S and the eukaryotic ribosomes are 80S? This is actually really important. The constitutions of our ribosomes are critical. This is actually how a lot of antibiotics, like say tetracycline or any kind of aminoglycoside, these antibiotics work because they target only the bacterial ribosome. And so because the bacterial ribosome is sufficiently different enough, and in one of those ways it's because of the Vedgar coefficient, it's sufficiently different enough from our own eukaryotic ribosome so that these antibiotics don't hurt us, but they will indeed harm the bacteria infecting us. So actually, I think this, yep, I was right. Sorry. I think this is an image that kind of shows the difference between the ribosomes. So you can see here the prokaryotic one is a lot smaller, right, than the eukaryotic one. And again, just the corresponding small and large subunits are smaller in the prokaryotic than they are in the eukaryotic. In addition, they also have specific sequences to them. So the small subunit in bacteria has a 16S rRNA. The large subunit has 2 rRNAs, a 23S and the 5S rRNA. So, and this is important, eukaryotes, we have different kinds of rRNA. So 18S in the small, 28S, 5.8, and 5. It's actually really interesting. This is kind of, I'm digressing a little bit, sorry. But you guys know how I like to go off topic just a tad. This is how we figured out that archaea existed in the first place. So you know that archaea are types of prokaryotes, right? We initially had put them together with bacteria, but then we saw that there was a specific group that we thought were bacteria. It turns out that they were archaea. This group actually had different ribosomal sequence to the ribosomes were completely different. And ribosomes are really key critical features in cells, right? They're the sites of protein synthesis. So they don't really mutate very often. You want to keep them as, you know, the same as possible, because any kind of mutation that happens there could be very problematic to the species, right? So they don't mutate very often. And so the fact that this other completely new group had another kind of ribosome was a big deal. And in fact, their other kind of ribosome was more closely related to the eukaryotic ribosome than it is to the actual bacterial ribosome. For example, archaea actually have a 5.S rRNA, which is mind-blowing. So their ribosome is actually more similar to our eukaryotic one than the bacterial ones. We obviously have a lot of differences, especially if you think about the proteins involved as well. Yeah, they have more proteins that are similar to ours than they have that make up their ribosomes, than they have proteins that are similar to prokaryotes that make up their to bacteria that make up their ribosomes. So even this image in itself is actually a little bit incorrect. Instead of saying prokaryotes here, I would say bacteria, right? Because prokaryotes also includes archaea. But this is pretty much bacterial ribosomes are 70S, and they have these particular constituents about them. And eukaryotes are 80S, and they have these particular constituents about them. So archaeas are a little bit difficult because we still don't know that much about them, but we're learning more and more each day. So so basically a ribosome is made out of rRNA and proteins. this rRNA is obviously something that is essential, right? And it's coded by our DNA. Remember that DNA can stop at RNA, right? It doesn't necessarily have to become mRNA and then go into protein. It can stop and be like, there's genes that encode for specifically just rRNA, or genes that encode for tRNAs. And so it can stop right there after transcription. And since they're so important, usually in an organism, we have a lot of genes. It's very redundant. We have a lot of genes that encode for our RNA. The genes, so the DNA that encodes for our RNA is known as rDNA. So that's how I'm going to be referring it from now on. So there's a lot of redundancy in our rDNA. In our rDNA. That is very difficult to say. Our rDNA. Oh my goodness. Okay, so an example of this is, if you guys remember back when we were talking about Down syndrome, how we had that case of familial Down syndrome, right? And how a person who actually was lacking some of their chromosomes, so some of their chromosome 21, remember that Robertsonian translocation that we talked about, how they had a little bit of 21 attached to a little bit of 14. And so they were both missing pretty much the P arms of both of those chromosomes. And yet, even though they were missing chunks of the chromosomes, they're still okay. Like, they still look normal. They aren't symptomatic at all, right? But they're potential carriers. They are carriers, and they can pass and give familial Down syndrome to their offspring. Now, that region that they're missing is not causing any problems, because that region is packed full of rDNA. And one thing that organisms have is that they have rDNA everywhere. Right? There's a lot of rDNA around it, like found within the genome of an organism, just because it's such an imperative thing. It basically codes for one of the structures of the ribosome, which is an incredibly important critical feature, right? So it's very repetitive, and it can be present in clusters at various chromosomal sites. Each cluster contains tandem repeats separated by non-coding spacer DNA. So chromosome 21 is actually one of those chromosomes that has, like, the P arm of chromosome 21 has those clusters of rDNA. So does chromosome 13, 50. There's a bunch of different chromosomes out there that basically have a lot of rDNA. And so these people don't really have that many symptoms, because even though they don't have a little bit of that rDNA, they have other genes and other chromosomes that can encode that rDNA. And the same can be said about other organisms as well. Bacteria have a lot of these different clusters of rDNA. In fact, actually, bacteria, we looked over in chapter 12 how they have polycistronic mRNA, right? And so what they actually do there's 3 types of rRNA. So they have 16S, 26S, and 5S. They actually just make it in one mRNA. Like, all these genes, the rDNAs that code for those rRNAs are found are found right next to each other, in a cluster right next to each other. So when the mRNA is made, all 3 are transcribed in one mRNA, the polycistronic mRNA. And then once the mRNA is made into a polypeptide, once we have that mRNA, there's no further point, because we're done. We're arrived at the RNA. At that point, it just gets cut. Those 3 get cut and separated, and we have our 3 types of rRNA, which is pretty phenomenal. So, and then these things are repeated a lot, right, throughout the genome, just because, again, very important. So then we move on to tRNAs. So these are our transfer RNAs. This is what actually is transferring our amino acid to the actual, the synthesizing site, the protein synthesizing site. So these are quite small. They're 75 to 90 nucleotides long, and they're very stable. They're stable because the RNA isn't really completely single-stranded. It's not linear. They kind of wrap around it themselves. They kind of form these loops, and within these loops, they have complementary. The loops are formed in the first place because they have complementary sequences between them that bind together. So they contain a lot of post-transcriptionally modified bases that are unique to tRNA. We actually went over this. If you remember, after transcription, there was instances where we have modification of bases, or things like RNA editing, right? Where a base could be changed specifically and made into something else. One thing actually that's unique to tRNAs is the base inosine. Do I talk about inosine? Hold on, I don't remember if I talk about it. Oh, there it is. I do. Okay. Oh, there's other ones as well. Okay. There's a lot of bases, so nucleotides in tRNA that aren't really conventional. So inosine is a big one. Pseudouridylic acid is another one, kind of similar to uracil. That's kind of like fake uracil. There's a lot of different kinds and kind of spin-offs off of these bases. And inosine comes about through RNA editing. Usually, specific adenines within the RNA get changed into inosine. And the interesting thing about inosine is that it can bind to adenine, so it's complementary to adenine, cytosine, and uracil. So it's just adding to that extra wobble hypothesis. You guys remember the wobble hypothesis? How the third, the last of the third the third ribonucleotide in a codon doesn't necessarily have to bind perfectly to the anticodon, to the complementary anticodon ribonucleotide. And inosine is one of them, right? Inosine, since it can bind to so many different ribonucleotides, it basically adds on to that wobble hypothesis. So it's very important to remember that we have post-transcriptional modification, especially when it comes to tRNA. We have a lot of unique bases in tRNA. So that's kind of one of the reasons how we can explain inosine. But for other bases, it gets a little bit more complicated. We don't really understand why there's so many different kinds of unique bases in the picture. And they say here, the presence probably enhances hydrogen bonding efficiency during translation. Okay, so when we look at our tRNA, there's two different ways that we can actually look at the structure of our tRNA. We can look at it on like one plane. So if we just basically get our sequence of RNA and then allow it to bind to each other, like, the complementary places to bind together, this is just basically a two-dimensional model. It's one plane. We see a clover leaf. It basically takes up the structure of a clover leaf. And you can start seeing where the anticodon is, right? This is the anticodon. You have the 3' end and the 5' end. The 3' end is where our amino acid is going to bind, right? And it's usually, the 3' end is usually, the terminal end is an adenine followed by a cytosine and another cytosine. So I don't know if you can see, it's really tiny. I can barely see it. Yeah, there we go. And adenine is our last one. This is so basically our amino acid gets bound to this adenine, right? To the hydroxyl group of the adenine. And then we have a cytosine and another cytosine. The 5' end has a guanine. So that's basically the same for every single tRNA out there. When we actually go ahead and look at the 3D configuration, so the actual proper 3D shape of this tRNA, we can see that it forms an upside down L, kind of like the one that I attempted to draw before. And then this upside down L is really nice because we can see like the actual loops forming places, and it's just the different domains that we find on the tRNA. So one end is the anticodon right here, this end right here, this is the anticodon. And the other end, you have the 3' end, which is where we just saw, we have that adenine that binds to the specific amino acid. So we have an anticodon loop. This base pairs with the complementary codon on the mRNA. In the picture, oh, cool. In the picture, I actually didn't even notice that. In the picture, you can see that actually it gives you inosine as one of the bases, right? So the anticodon here is CGI. And so remember, this inosine can then go ahead and bind to a lot more bases than typically other nucleotides can, other ribonucleotides can. It can bind to U, C, or A. Oh, good. I didn't realize it was written on here, so this is really good that it's written. Okay, so in the picture, you can see inosine. It can base pair with U, C, or A, and it basically makes for more availability, more codons to be recognized because it can bind to more of them. So all anticodon loops are present in the same position of the clover leaf, and therefore, and also of the actual three-dimensional figure, right? It's always it's the opposite end of the 3'. So the corresponding amino acid is covalently linked to the 3' end of all tRNAs, to the adenine, to the hydroxyl of the adenine. The tRNA 3D structure is roughly L-shaped, with the 5' and 3' ends both located near one end of the structure. Yeah, and then the protruding A residue, the adenine residue, is the attachment site of the amino acid. So it's kind of all repeating itself a little bit. Okay, here you can see just the different kinds of, like the secondary structure, that 2D one plane model, and then the actual tertiary structure, or the 3D configuration of the tRNA. This place, so you see the CCA, this adenine is the 3' end where we would normally add our amino acid. It's also known as the acceptor stem. And then on the opposite side, we have the anticodon loop, which, as its name suggests, holds the anticodon, which is going to be complementary to the actual codon, right? So an important thing about tRNA is that obviously it has to transfer the amino acid. So the amino acid has to be loaded onto the tRNA. And what we call this loading process, it's the same as saying charging the tRNA. That's what we use. We charge the tRNA. This actual reaction is known as amino acetylate. Nope, I screwed it up, sorry. Amino acylation. I was about to say acetylation. Nope. Amino acylation. And it's carried out by the amino acyl tRNA synthetase enzyme, okay? Amino acyl tRNA synthetase enzyme. I'm probably going to call it amino acyl synthetase because it's the same thing, okay? So this is an enzyme that will load the appropriate correct amino acid. It basically, the enzyme takes into consideration the anticodon of the tRNA. It knows what that anticodon, what codon the anticodon is complementary to, and it will figure out which amino acid corresponds to that codon. And it will charge, remember that word charge, it will charge the tRNA with the appropriate amino acid. So it's the enzyme that catalyzes the amino acylation reaction. It's highly specific. It recognizes only one amino acid and then subset of tRNAs with their subset of anticodons that are complementary to that subset of codons that code for that amino acid. Because remember, the genetic code is degenerate, right? And so sometimes we have multiple codons that code for one amino acid. So if you remember, leucine has 6 codons that code for one amino acid. This would mean that the amino acid tRNA synthetase that can basically charges a tRNA with leucine is able to recognize all the tRNAs that have the anticodons to those 6 codons that encode leucine. And to those 6 anticodon tRNAs, we add, or the amino acyl synthetase adds on the actual leucine amino acid. Okay, accurate charging, as you can imagine, is extremely crucial if fidelity of the translation is to be maintained. So this is a big point. And in fact, it's extremely specific. There's not a lot of mistakes that happen here, thankfully, because if there were, we'd be in a lot of trouble, right? So there's a synthetase, there's an amino acyl synthetase for each amino acid. So there's 20 amino acids out there. We have 20 amino acyl synthetases. Each one is going to load one specific amino acid. And then there's about 31 tRNAs, just because remember of that wobble hypothesis that we considered even like in the prior chapter. There's not 61 synthetases, again, like there's no need to make 31 synthetases either, because one synthetase can recognize multiple tRNA anticodons, just as long as those anticodons are complementary to the codons that code for the same amino acid. So the specificity of the amino acyl synthetase all has to do with the amino acid, okay? Yeah, so the charging process, as you can imagine, requires a lot of energy. So it is basically spurred on by the use of ATP. So we have our amino acid, as you can see here, and then ATP. The amino acid is converted to its active form by reacting with ATP, and this creates amino adenylate acid. So it basically, the adenine and one phosphate gets added to the amino acid, and then we release 2 phosphates. And that release of those 2 phosphates actually kind of spurs on this reaction. It's the removal of a pyrophosphate, that's what we have 2-phosphate, helps, like it basically powers us through the reaction. This activated form of the amino acid remains associated with the enzyme until the enzyme actually interacts with a specific tRNA. So at this point, the enzyme is holding the amino acid, the activated one, and then once it gets a tRNA that holds the correct anticodon that is complementary to the codon that codes for the amino acid that it's holding, it will then load or charge the tRNA with the appropriate amino acid on the 3' end of the tRNA, right? So it will basically transfer, the amino acid is going to be transferred to the appropriate tRNA, sorry, and bonded covalently to the adenine residue at the 3' end. And so once our tRNA is charged, this is now known as an amino acyl tRNA. And usually we don't even call them amino acyl tRNAs, we just call them by whatever amino acid they're actually holding. So a methionine tRNA or a leucine tRNA, whatever specific amino acid we are considering. Okay, so that's the end of section, I believe it's just 13.1. And I'll see you guys in the next one.